Babies. These are babies. Tiny, adorable creatures that are remarkable bundles of potential, full of innocence, full of wonder. And in the very cells of babies are genes. Genes that will determine their characteristics as they grow to be fully functioning adults. Their eye color, hair texture, height, and most importantly, their tendencies and susceptibility to certain health conditions.
That said, I do not want my babies to inherit my diabetes or to be susceptible to heart diseases. What if I tell you that we now have the capability and the technology to edit human babies, specifically the human genome, or what we may call it, gene editing? Today we are going to continue tackling the book entitled
The Song of the Cell, an exploration of medicine and the new human, by Dr. Siddhartha Mukherjee, and we will dig deep straight onto the next chapter entitled The Tampered Cell, Lulu, Nana, and the Transgressions of Trust. In the previous chapter, we were introduced to the concept of in vitro fertilization or IVF, a technique that involves the fertilization of an egg by sperm outside the body, typically in a laboratory setting.
Although IVF was predominantly used by individuals or couples who face challenges with conception, advancements in research made it possible for the application of genetic, reproductive technologies, and even gene editing in conjunction with IVF, engineering our way towards greatness or of possible destruction.
In a quote from the British embryologist Ian Wilmut, it is difficult to imagine a greater imposition than adding genes to future generations that changes the nature of future people.
With this, Wilmot highlights the possible contributions of genetic engineering. However,
In a letter by the astronomer Martin Ryle to the Pontifical Academy of Sciences, he stated that the benefits of medical research are real, but so are the potential horrors of genetic engineering and embryo manipulation.
really, genome editing is a truly wonderful field of science, but it can be very scary very fast. And that's exactly what we are going to focus on today.
We are going to learn about the brief history of reproductive biology, cellular medicine, and gene editing, the tragedy of Hajan Kui, some takeaways we can use for our course, biology, and the importance of ethics, trust, and transparency in conducting medical procedures.
Especially procedures that involve human lives.
On June 10, 2017, Ha Jiankui, the infamous Chinese biophysicist turned geneticist who goes by the nickname JK, wanted to perform an IVF medical procedure on an embryo with the intention to permanently alter the embryo's genes and essentially create the first ever transgenic or gene edited human.
It truly is shocking, I know. Even the whole scientific community got flabbergasted by the news. But why? Before we actually discuss the turmoil brought by Haj and Kui and why it was so controversial, let us first sort out the short history of reproductive biology and the birth of cellular medicine through the lens of this chapter. To fully understand why and how Haj and Kui came up with the idea to edit the human genome.
In 1968, Robert Edwards of IVF fame had taken interest in reproductive biology and published a paper regarding sex determination in rabbit embryos and was successful. Edwards utilized the dye chromatin to figure out which cells from the blastocyst have the X and Y chromosomes, thus developing a genetic assessment based embryo selection.
Although this was not Edward's initial thought. Instead, it was sparked by his curiosity whether chromosomal problems could be detected in the embryo. such as the problems with the chromosome 21 for Down syndrome, and whether they can be discarded. Such situations became one of the primary focuses of subsequent research.
Not long after, by the 1990s, advancements in IVF and genetics allowed Edwards' method to be tried on human embryos. Spearheaded by the scientist Alan Handyside from London, he worked with couples with a history of X-linked diseases, in which only male children are at risk. Handyside was able to ensure that only female embryos will be implanted. Birthing the technique, pre-implantation genetic diagnosis, or PGD.
Basically, couples can now choose whether they want a male or a female baby. However, embryo selection is a negative process, which means that it involves the removal of embryos. But that also means that we have the possibility for a positive process, which can be used to resolve issues and diseases that require either the addition or alteration of genes.
In 2011, a prominent focus emerged on a gene-altering technology known as gene editing. And by 2012, leaders in the field of gene editing, such as Jennifer Doudna, Emmanuel Charpentier, Feng Zhang, George Church, and many others were able to discover one of the greatest advancements in genetic engineering, the CRISPR-Cas9. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats, which is a method derived from bacterial defense systems, while Cas9 is a search and destroy eraser protein utilized for deliberate and specific alterations, often involving targeted gene disruption through precise manipulation or cutting.
However, this groundbreaking technology is still far from perfection and may have occasional errors we know as of target edits. Basically, these are unintended gene modifications, or mistakes as we may call it, that may cause unknown and unwanted effects to the host, a risk in the field of gene editing. Now that we have learned the brief history of reproductive biology, cellular medicine, and gene editing, let us have an in-depth discussion about
The Tragedy of Ha-Jen Kui.
gene editing has been long since a controversial and extremely ethically complex topic. The idea of gene-edited babies gained global attention when the Chinese scientist claimed in late 2018 that he had created the world's first gene-edited babies using CRISPR-Cas9 technology. Impressive, right? I mean, those are living, breathing, gene-edited or transgenic babies. But why is it a tragedy rather than an achievement we should all celebrate? The answer lies on the fundamental criteria of gene editing. The experiment must be well thought and must follow a critically designed procedure. For starters, to be able to conduct gene editing in humans, one must first reach certain requirements and answer questions related to safety and ethical issues.
Does the researcher have informed consent from the participants? Was it achieved in a moral and human way? Is the methodology of the procedure acceptable and precise? Can they ensure the safety of the baby? Will the experiment yield desirable effects or address certain medical issues? Will the researcher maintain a regulatory oversight and follow strictly the given ethical guidelines? Does it have approval? Probably by an institutional review board.
Only then, once reviewed and given a go signal, can Hajan Kui proceed with this tiny little procedure. But nope, he decided to skip all odds and get straight into it. Hajan Kui could be called a living embodiment of the inverse of the proverb, do first, think later. Well, enough introduction, here's how the story goes.
First, JK claimed to have received approval from the Medical Ethics Committee of Shenzhen Harmonicare Women's and Children's Hospital for his gene editing study in human embryos. However, investigations revealed that the hospital denied granting approval or even having documented evidence supporting JK's claims. This discrepancy raised significant ethical concerns about the experiment's ethical foundation and regulatory oversight. The lack of authenticated approval highlighted the potential dangers of conducting sensitive experiments without
In
proper
short,
institutional scrutiny
Hagen's
and validation.
experiment was doubtful and doomed at the outset.
Nevertheless, he continued with the procedure wherein the gene targeted by JK was CCR5. Now, some people have the Delta 32 mutation, which is a mutation that involves the deletion of 32 base pairs within the CCR5 gene, leading to a structural change. This was a notable mutation since the individuals who carry two copies of the Delta 32 mutation, one inherited from each parent, are highly resistant to HIV infection.
JK aimed to mimic the Delta 32 mutation naturally found in the CCR5 of some individuals to explore the possibility of conferring HIV immunity to individuals through genetic manipulation.
Next, the couples involved in the study were selected by JK because the father had a chronic but controlled HIV infection. However, the risk of HIV transmission after the sperm was washed pre IVF by JK himself was extremely low, actually zero, in this scenario. Simply put, he made a mistake with his parameters. Thus, these embryos face no higher risk of HIV infection than embryos conceived by a couple not affected by HIV.
Additionally, editing the CCR5 gene, a gene crucial for immune response regulation, posed unforeseen effects, such as being at more risk for other viruses like the West Nile and influenza. This sparked ethical discussions regarding the experiment's legitimacy and potential unnecessary harm to the embryos.
The experiment involved a pretty standard IVF procedure. However, there is an introduction of the CRISPR-Cas9 system through intracytoplasmic sperm injection or ICSI, aimed to edit the CCR5 gene in the embryos. This precise genetic manipulation method highlighted the technical advancements in gene editing, but also raised ethical concerns about the possible irreversible alterations made to the embryos and to the future offsprings.
After 6 days, JK reported that only 4 zoozygotes successfully grew into viable blastocysts, while being successful in editing only 2 out of the 4, leading to the birth of the twins, well, from the title, Lulu and Nana. Well, it's not the real name for the sake of confidentiality.
However, the gene edits did not exactly replicate the intended mutation, but instead introduced novel genetic variations. These alterations are different from the naturally occurring Delta 32 mutation, raising uncertainties about the health implications for the twins. Additionally, there was a possibility that only some and not all of the cells have the Delta 32 mutation, inducing the twins in the phenomenon that we call as mosaicism.
The emergence of these unknown genetic variations, along with the potential of target edits, will, of course, undoubtedly, spark ethical debates regarding the unforeseen risks and consequences of gene editing on human embryos.
Well, the news about Hagen-Kruse experiment made the press, and at the International Summit on Human Genome Editing, JK was introduced by the geneticist Robin Lovell-Badge to share his procedure and results. Now, contrary to his expectations, JK faced extensive questioning and scrutiny about the experiment. The attendees raised ethical concerns about the informed consent procedures, the potential health risks to the twins, and the ethical framework guiding the experiment.
The lack of clear ethical guidelines and uncertainties surrounding the health implications for the twins triggered a global ethical uproar within the scientific community, emphasizing the need for stringent ethical oversight and regulatory frameworks in conducting genetic experimentations on humans.
JK was also not able to answer the simplest questions regarding his results. To top it off, Lovell Badge himself questioned the potential risks of gene editing on Lulu and Nana, reiterating the validity of consent and the recruitment process on the two couples. However, JK's response has remained vague. In response to the painfully obvious ethical uproar and the potential dangers highlighted by JK's experiment, Ha Jiankui faced severe criticism and legal consequences. In late 2019, he was sentenced to 3 years in prison and was barred from conducting any future IVF research.
This case serves as a stark reminder of the intricate ethical dilemmas and potential hazards associated with gene editing on humans, specifically experiments that do not have a proper oversight and a transparent ethical considerations, prompting a reevaluation of ethical boundaries in genetic experimentation.
Now let's set aside for a few minutes the term will caused by Ha Jiankui and focus on the geneticist Denis Weberkov. In June 2021, Denis Weberkov, a passionate geneticist from Russia, revealed plans to edit the GJB2 gene associated with the hereditary human deafness, wherein inheriting two mutated copies of this gene leads to deafness, a tone eerily similar to how Hajankwi started. But Rebrakov, aiming to follow pioneers like Steptoe and Edwards, considers himself a cautious trailblazer. Despite intending to adhere to strict regulatory approval and informed consent standards, he remains committed to proceeding with embryo genetic manipulation.
Robrikov's approach involves a methodological progression, sharing data through publications, conducting comprehensive genome sequencing to evaluate on-target and off-target edits. He emphasizes that his therapies will cater exclusively to deaf couples with dual copies of the gene mutation who provide full consent and aspire for a non-deaf child. Hajan Kui is a total opposite of Rebrikov, someone who is well prepared, respects the code of ethics and transparency of such experiments, someone who thinks before they act. Looking back at the history of gene editing, its applications and the experiment of Hajan Kui, we can pick bits by bits of information and learning from the aforementioned topics and integrate it into our own understanding, creating a whole new different mode of perception that we can tie back to our curriculum and to cell and molecular biology.
Cell and molecular biology is the backbone for understanding basic chemistry and biochemistry, explaining how molecules such as DNA, RNA, proteins, and lipids function within cells, providing insight into the chemical basis of life. In terms of pattern inheritance, by studying cells, genes, and how they work, we get important clues about why we look the way we do or why some diseases run in the families. Additionally, through advancements like the CRISPR-Cas9, students learn how tiny changes in our genetic material are made and how gene editing affects us. It's all about learning how to edit our genes to help in medicine or research. Inherently, there are certain topics in cell and molecular biology that are directly tied with the chapter. Such topics include recombination and recombinant DNA, IVF, and gene editing. Firstly, cell and molecular biology help mix and match genes from different sources to create new DNA sequences. This is done by understanding how cells work and using techniques to change genes in a lab. It is definitely useful in procuring important inventory like medicines and improving crops. For IVF, by knowing how these cells behave, we can do procedures like collecting eggs and fertilizing them. They also use techniques to check which embryos are healthiest before placing them in the womb. Lastly, cell and molecular biology play a big role in gene editing. Through CMB, we learn and understand how genes work and utilize tools like the CRISPR-Cas9 to edit DNA, potentially fixing genetic issues or altering traits for medical purposes.
Harajanku's decisions while conducting gene editing reflected a series of missteps: the choice of incorrect genes, inappropriate candidates, and the flawed procedures. His motivation stemmed from the desire to pioneer a new technology, seeking the coveted status of being the first. Yet, it is critical not to absolve him of his actions, of his choices, considering that other scientists at his time with access to the similar tools were able to exercise restraint. Well, nevertheless, Jan Key's actions revealed the beautiful intersection of gene editing and assisted reproductive technologies, showcasing their potential for scientific progress while uncovering significant ethical challenges. Moreover, beyond the immediate concerns about gene editing, the case prompted broader reflections on its implications for in vitro fertilization or IVF and reproductive technologies. It highlighted the critical importance of reevaluating and reinforcing the ethical guidelines within IVF procedures to ensure that there is responsible and ethically sound use of these emerging and limited technologies, prioritizing the welfare of future generations. Looking ahead, Junka's case serves as a crucial lesson for everyone, for scientific research and further technological advancements. It calls for a unified commitment among scientists, policymakers, and stakeholders to establish clear ethical boundaries, robust regulatory frameworks, and, of course, transparent communication channels. This collective effort isn't just about advancing scientific frontiers, it's about upholding eth- ethical integrity and fostering societal trust, while navigating the uncharted territories of genetic technologies, avoiding another case of Lulu and Nana. Before we officially wrap this chapter, let me share with you a short prelude for the next chapter, the developing cell. Starting with a question to ponder on:
Why did we ever leave the single celled world and chose to form multicellular organisms?
the process of development, starting from a single cell to complex embryo to a human being or another organism, raises question about how multicellular organisms began.
Biologist Nick Lane highlights similarities between cells in simple organisms and those in complex ones, igniting curiosity about why this shift from single cells to complex life happened. A millennia ago, formations resembling fern fronds emerged, marking the inception of multicellularity in blue-green and green algae. Sponges formed from amalgamated individual cells, while microorganism colonies heralded a new existence.
Remarkably, multicellularity independently arose in various species, echoing Richard Grossberg and Richard Stratman's notion of a minor major transition.
A compelling experiment led by Michael Travisano and William Radcliffe at the University of Minnesota showcased the evolution of multicellular entities from unicellular yeast. Through successive rounds of selection and growth, these researchers cultivated yeast clusters exhibiting a novel, crystal like structure, an evolution into multicellularity that resisted reversal.
Astonishingly, these clusters reproduce by division and develop a mechanism involving programmed cell death, fostering detachment and individuality, showcasing the rapidity in which these single cells, can evolve into multicellular aggregates. Prevailing theories suggest, though, that specialization and cooperation optimize energy and resources, propelling evolution towards collective survival. This intriguing evolutionary trajectory hints at a purposeful and directional evolution, marking a pivotal point where these clusters might eventually be deemed as fledgling organisms. And that's it, folks. We have successfully discussed the brief history of reproductive biology, cellular medicine, and gene editing, the tragedy of Hagen Kui, and the importance of ethics, trust, and transparency in conducting human associated procedures. As always, thank you for watching and join us again on the next video where we will discuss how a simple cell develops to be an organism. Have a nice day and remember, don't stop learning.